Yes, hi everyone, Yugi. Having looked at mitosis, I want us to conclude by this process of cell division by looking at meiosis. Yes, meiosis. So meiosis, as in simple terms, you can say that this is a type of cell division in which the diploid parent cell divides into 4 daughter cells, each having half the number of chromosomes as the parent. So you are seeing that a diploid cell, that is parent cell, which is diploid, is dividing us, giving us haploid daughter cells. That is, one of it is giving us 4 daughter cells, but these 4 daughter cells, each is having half the number of chromosomes as that of the parent. Understand? So that is meiosis. So meiosis takes place in testes and ovaries of animals, and in plants it occurs in the anthers and ovaries. So you see that this meiosis, so this signifies that this meiosis mostly occurs in reproductive parts of both plants and animals. That is, reproductive parts in animals, you are seeing that it occurs in the testes and ovaries. Test, that is in male, and ovaries in females, whereas in plants it occurs in anthers and ovaries. So from the diagram we are seeing here that meiosis is a diploid parent cell divides in meiosis 1 because it has meiosis 1 and meiosis 2. So in meiosis 1, a diploid parent cell that is 2N divides in meiosis 1, giving us 2 daughter cells. So the 2 daughter cells undergoes meiosis 2 and form what we call 4 haploid, that is N progen cells. After second meiotic division, it forms the progen cells, which are 4, each cell having half the number of chromosomes as that of the parent. So this is meiosis. It has 2 phases: meiosis 1, or first meiotic division, and meiosis 2, that is second meiotic division. As we know that the second meiotic division is always the same as mitosis, yes, as we shall see. So meiosis, also known as reduction division. Reason being, meiosis, we are seeing that a diploid parent cell, which is diploid, give us 4 progen cells, each having half the number of chromosomes as that of the parent. So the chromosome number has been reduced to half. So that's why it's called reduction division. Meiosis consists of 2 successful division, that is first meiotic division, where the parent divides into 2, and the second meiotic division, where the products divide to produce 4 daughter cells. So the process involved in the 4 stages, that is of meiosis, we have as prophase, metaphase, anaphase, telophase, but distinguished by meiosis 1 and 2. Yes, it's distinguished by meiosis 1 and 2 for the first and second meiotic division. That is for the case of meiosis 1 and meiosis 2. Both of them will have the prophase, metaphase, anaphase, and telophase stages. Yes, as we shall see. So we know that mostly we shall start with the first stages of meiosis. We shall see that we shall have interphase also involved. So this diagram here is a diagram of DNA. So before you go to interphase, I request you first follow, like, share to your fellows, and make this video move so fast, so as we help each other. That is in the fact. So let's start with the interphase stage of meiosis. So you're saying this is a DNA. So that the structure of DNA is a double helix. These ladder-like structures you see, this is the DNA. These are base pairing. So these are base pairing. So you say the DNA is double helix. It's 2-stranded. It has 1 strand and the other strand is called double-stranded DNA. So these are called the bases. So in DNA, The bases we have A, adenine, cytosine, guanine, thiamine, that is ATGC. This is just a simple structure, so maybe you shall find sometime discuss about the DNA and protein synthesis, but for now let's continue with interphase. So interphase, I see that in interphase of meiosis, chromosomes not visible, so they are still thread-like. That is interphase. Chromate is present, duplication of DNA, production of ATP. So we say that duplication of DNA occurs in meiosis, but we shall say that in meiosis 2, there will be, before the cell, after meiosis 1, the cell will not undergo DNA duplication again. It will duplicate only in meiosis 1. So we are saying that there is duplication of DNA deoxyribonucleic acid will replicate, production of ATP, energy will increase because we need energy in movement of chromosomes, formation of new organelles will be formed. For example, centrioles, that is in animals, will be formed, and other organelles like the mitochondrion, that is for energy. Yes, centrioles are the formation for formation of the spindle fibers. We know that they form the esters. The esters which protrudes and form the spindle fibers, where the centromeres of the chromosomes are arranged as they move towards the equator, as we shall see. So let's start with prophase 1, that is in meiosis. So in prophase 1, the nucleolus disappears. Yes, the nucleolus disappears. Centrioles arranged on the opposite sides of the nucleus. Spindle fibers, they start to form. Chromosomes condense. Say that in interphase they are still thread-like, but here they start to condense. That is the chromatid now condense to form the chromosomes. Homologous chromosomes come together. So that process of homologous chromosomes coming together, it is called synapsis. Synapsis is that situation where the chromosomes now come together, pair up with one another. That is called synapsis, forming a bivalent. So a pair formed by synapsis is called a bivalent. A bivalent is when they are now in a pair, two of them. Yes, that is at the prophase. As we are seeing the diagram, homologous chromosomes forming the bivalent. So the bivalent are these two pairs of homologous chromosomes. After synapsis has occurred, form the bivalence. So this is the prophase 1 of meiosis. Let's go to metaphase 1 of meiosis. So in metaphase 1, homologous chromosomes move to the equator of the spindle. Yes, they move from the opposite poles to the equator of the spindle. So behaves as a unit. So they move in as a unit from the opposite poles to the equator. So the chromosomes arrange themselves on the equator of the spindle, as we are seeing this diagram here. You can see from here that they arrange themselves to the equator of the spindle. That is the mostly metaphase. That is what you call. We are just giving a summarized way of how we can go about this meiosis. So this is what occurs in metaphase. So in anaphase 1 of meiosis, that is homologous chromosomes part company or move towards opposite poles of the spindle. Part company or move towards opposite poles of the spindle. These homologous chromosomes part company and migrate to the opposite poles of the cell. So they tend to move from the equator back to the opposite poles of the spindle. That is the homologous chromosomes now. So in telophase 1 of meiosis, we are seeing that for this case, the chromosomes have reached their destination and the cell constricts across the middle as in mitosis. So you see that the chromosomes have reached their destination, that is the opposite poles, and they start to the cell constricts across the middle as in mitosis. So it constricts like it tends to to form a furrow or a cleavage. This is in the middle, as we are seeing the diagram here. The first meiotic division results in two separation of homologous chromosomes. So our first meiotic division will give us result in two separation of homologous chromosomes. So this is the first meiotic division. So after the first meiotic division, the cell now enters the second meiotic division. But before that we go to second meiotic division, that's a simple summary of what we can talk about. That is in meiosis 1. But somewhere, let's have this summary again. You want to leave someone behind. So interphase, for interphase, say that the chromosomes are still invisible. So the chromatin, we have DNA replication, production of ATP, that is interphase. Prophase 1, you have nucleus disappears, centroids arranged on the opposite side of the nucleus, spindles forms, chromosomes condense, homologous chromosomes come together, that is from synapses, forming a bivalent. So in prophase 1, that is where crossing over occurs. Crossing over of sister chromosomes now occurs. Crossing over, that is, these sister chromatids tend to repel each other. So they form, they tend to exchange. One chromosome and the other tend to come together and exchange the contents. So they form that point where they meet. It's called the chiasmata or the chiasma. So at chiasmata, that is a crossing over point where exchange of materials occurs between one chromosome and the other. So at chiasmata, that point, understand, that is where crossing over occurs. That is prophase 1. Yes. Metaphase 1, say homologous chromosome move to the equator of the spindle together as they move from opposite poles to the spindle. Anaphase 1, homologous chromosomes part compound or move towards the opposite poles. Chromosomes are now at what compound are going to the opposite pole, that from the equator. And telophase 1, finally, we are saying that the chromosome have reached their destination and the cell constricts across the middle. That is the summary. So we go to second meiotic division, but if you go there, make sure that let's like, share, follow, and comment as we go for the second meiotic division. So the second meiotic division, let's look at the second meiotic division in the next video.